Hello everyone. Today we are going to discuss the very important topic for Gpat and NIPER point of view, and the topic is a cholinergic drug. And in this video, I will cover the synthesis of acetylcholine, then SAR of acetylcholine, then mechanism of acetylcholine and destruction of acetylcholine. And let's start. That is a cholinergic system. Okay, cholinergic system. Cholinergic system. Firstly, we discuss the synthesis of acetylcholine. Okay, for the synthesis, we require the source of energy called as ATP, then inactive acetate. Okay, then enzyme called as coenzyme A. Okay, these three are the precursor for the synthesis of acetyl coenzyme A. Okay, acetyl coenzyme A precursor, and this reaction we called as a acetate activating reaction. Acetate activating reaction. Then this acetyl coenzyme A convert into the acetylcholine. Okay, acetylcholine by using the enzyme called as a choline acetyl transferase. Okay, this is a choline. By using choline plus acetyl coenzyme A plus choline is a formation of acetylcholine. Okay, now we will see the structure of acetylcholine. This is the structure of acetylcholine. Okay, in this structure there is a two center. This is the cationic and this is a salt of this acetylcholine, Cl minus. Okay, and this is the quaternary ammonium. Okay, quaternary ammonium. This is the alkyl chain. Okay, this is the alkyl chain, and this is the acetyl group. Okay, acetyl group. Now we will see the SAR of acetylcholine. Okay, this quaternary ammonium binds to a anionic site. Okay, this anionic site, and this alkyl chain act as an van der Waals force. Okay, van der Waals force, and this acetyl group bind to the esteric site. Okay, esteric site. This is the synthesis of acetylcholine. Okay, synthesis of and this happen in the nerve end of neuron. Okay, now we are going to discuss the destruction of acetylcholine. Okay, see, this is the acetylcholine. It converts into the two. First one is a choline and then acetate. Okay, by enzyme choline esterase. Very important part for Gpat and NIPER also. They ask about the enzyme choline esterase. Okay, and this is the SAR. Now we discuss the effect of substituent on the this alpha carbon and beta carbon. Okay, this is the alpha carbon, this is beta carbon. Why we called it is the alpha carbon and this is called as beta carbon? Because alpha carbon are the carbon atom which are directly attached to a functional group. This is the acetyl functional group. Okay, in the classification of the cholinergic system, the functional group may be acetyl group or the functional may be carbamoyl group. Carbamoyl group just replacement of this CH3 group by NH2 group. This is called as a carbamoyl group. Okay, by replacement of this CH3 by NH2, there is a no change in activity. Okay, so we will see the what happened when we change the substituent on the CH to CH2. If we substitute the alpha carbon carbon atom, there is a then if we substitute this alpha carbon, okay, one is alpha, second one is alpha. If we substitute on alpha carbon, there is a decrease in a there is a decrease in muscarinic activity but increase in a nicotinic activity. What is a muscarinic and nicotinic activity? Means there is a binding of acetylcholine to the muscarinic receptor and binding of acetylcholine to the nicotinic receptor. These are the two types of receptor which are present at the end of neuron. Okay, there is the effector cell, we call it as a effector cell. Okay, so if there is a substituent on the beta carbon, okay, this is the beta carbon, there is an increase in muscarinic binding or muscarinic activity but decrease in nicotinic activity. Okay, and there is a no change in activity if we substitute this CH3 of acetyl by NH2, that is a formation of carbamide. There is a no change in activity. Okay, and this is the acetylcholine chloride. Okay, this is the structure of acetylcholine chloride. Now we will discuss the mechanism of acetylcholine. Okay, let's the all of you know that this is the nerve end. Okay, this is the nerve end, and there is a in this nerve end there is a packet of acetylcholine. This packet consists of this acetylcholine. This acetylcholine release at the end of nerve ending. Okay, this is acetylcholine release into the nerve end, and this directly attack on the receptor. This is a receptor. First one is a nicotinic, then this is nicotinic or muscarinic, any receptor. Okay, so this acetylcholine get converted and is converted into the choline and acetate by destruction. Okay, choline and acetate. Okay, by using enzyme the choline. This choline reuptaken through this is a there is a one channel we call it a sodium choline transporter. Okay, this is the sodium choline transporter. Okay, this is sodium choline transporter, and then choline is reuptake, and then again the synthesis of just like this acetylcholine synthesis happen in the nerve end. Okay, and they store into this that to prevent the reuptake of this choline, the drug called as a hemicholium. This drug prevent the reuptake of choline through a sodium channel, sodium choline transporter. Okay, see the release of acetylcholine also prevented by drug called as a vesamicol. Okay, vesamicol is a drug which prevent the release of acetylcholine through a nerve. Okay, this is the activity, this is the mechanism of action of acetylcholine. In the next lecture we will discuss the classification of drugs, means the drug which mostly used in the cholinergic system. Okay, so thank you for watching. Bye bye.